Hello, everyone. Thank you, Long Game, for inviting us to present in this demo day and for your continued support. My name is Oad Gafni. I'm the co-founder and CSO of Renewal Bio, and today I'm going to share with you how we're renewing human health. I would like to start with this painting of Lucas Carnas the Elder, the Fountain of Youth. It shows elderly, weak people and sick coming from one side of the fountain and coming out young, healthy, and ready to party. Unfortunately, we haven't found this fountain of youth yet, but we're making stride towards alternative. And there are quite a lot of evidence in the literature where blood from a young mouse was transfused into an old mouse through a blood circulation, shared blood circulation, and the young blood managed to rejuvenate the immune system, but not only the immune system, also the brain, the heart, and other tissues. And this set of experiments actually showed us that transplantation of young genetically matched cells and tissues can rejuvenate tissues and organs. And of course, a great promise to obtain young cells for various therapies came with the first derivation of embryonic stem cells. And for those who don't know, embryonic stem cells, the group of cells that later differentiate to any cell of your body and form the entire embryo. But there is a major roadblock mimicking the complexity of the bioenvironment in differentiating these cells. And that's why we created Renewal Bio and to basically address the problem to obtain young cells and tissues for transplantation. And this technology is based on an evolution of discoveries from Jacob Hana's lab at the Weizmann Institute in Israel. And we've created a new entity of therapy we coined as stemroids, which are human stem cell derived embryo models or developmental models from cells alone. So no eggs, no sperm, no fertilization, allowing authentic holistic differentiation to various cell types with just one platform. And this technology featured on the covers of Nature and Cell, the most prestigious science journals. And many in the scientific community believe that this discovery is actually a Nobel Prize worthy. And today I want to share with you a little bit on our tech and its market. So I want to start with our incubator, our roller culture that we've developed, which closely mimics the uterus environment, allowing embryos to grow outside of the uterus. This was a very complex goal, of course, because the conditions in the bioenvironments are very tricky to mimic. And when we took a mouse that was fertilized inside a mouse naturally, and we took the embryo to grow in our incubator, we saw that it develops properly, giving rise to all cell types, all tissues, all organs, as they normally happen in uterus. The development of this device allowed us to support proper embryo development outside of the uterus for the first time. And in this movie, you can see the gastrulation process. This is the most important process of differentiation, generating the various cell types and tissues that make organs. And as Louis Wolpert said, it is not birth, marriage, or death, but gastrulation, which is truly the most important time of your life. You can see here from these images that the embryo not only growing in size inside our incubator, but actually properly develops and creates the axis of development. You can see the head, the tail, the heart beating, the liver, and a lot of other organs, and just developing just like in utero. And this slide is a little bit scientific, but please bear with me because it encapsulates the whole process. So we start with embryonic stem cells or any other stem cells, and we transition them into our naive conditions. From these naive conditions, we can not only make the embryo proper cells, but we can make also the supporting environment of the extraembryonic tissues like the placenta, the umbilical cord, and the yolk sac. And when we aggregate them together in a special plate, we form these developmental models termed stemroids. And the models develop proper structure mimicking human embryonic development, as you can compare between the textbook image on the right here and stemroids on the 8th day of the protocol. You can see that they very closely resemble, and the stemroid actually mimicking the development. And the beautiful thing about our platform is as we develop further and as we move forward with development, we can make different cell types and we can basically isolate different cell types for different indication. And the only thing that differs in the platform is the way we isolate the cells. As we move forward with development, we unlock more and more cell types that we can use for different indication, and then later on tissues and organs that we can use. And this is an immense opportunity because the STEMBROI platform is super valuable because it helps us understand what's biologically possible. And then we can decide what makes sense clinically and economically. And our technology revenue potential is immense, of course. $117 billion only for the cell therapy market. But of course, it can be used as a progenitor cell source, platform for drug testing, and a lot more. Thank you for your attention, and I'll be happy to take any questions.
Thank you very much, Ohad. So yeah, this is definitely a very unique company in our portfolio, and kind of falls into what we would class as a category of kind of cell and tissue replacement. It's a little bit different than, instead of trying to slow down and reverse damage and aging in old cells and tissues, actually looking to replace them with new young cells and tissues. But of course, the challenge then is, how do you get those young cells and tissues? And that's what Renewal is really solving. So yeah, I think from our perspective, and fantastic scientific team, great investors and vision, and tackling that kind of challenge of how do you at scale make these cells. I know that, congratulations, since we invested, you've closed your latest sort of $5 million round, and have the firepower now to kind of move towards commercialization. So we'd be keen to hear more about how you're approaching that next step, especially the first cell type, HSCs. What does it look like technically and commercially, actually, to make those next steps?
Sure. Thank you, Will. So first of all, it was the seed round and it was 15 million. We just closed it. That was very exciting in this, unbelievable landscape. Yeah, so our first milestone is, of course, unlocking day 28 to 30, which captures actually a few cell types. But as you mentioned, hematopoietic stem cells are the first cells that we're going to deal with. Um, we chose the plastic anemia indications as bone marrow failure disorders. But as I mentioned in the talk, the hematopoietic stem cells are a great way to rejuvenate all tissues, especially the blood, of course, and dealing with pathogens and just rejuvenating the immune system and not only the blood. So, of course, that's our first goal, as you mentioned. But the beauty about our platform is that we're getting to day 28 and 30, and that's what we're doing right now. We're actually unlocking the different stages, and we're understanding that there are a lot more cells that we can actually use. We found out, about a few months ago, that endothelial cells are they have a market for that as well, for vascularization. So we're actively testing, and we're seeing that these cells are actually functional and they're forming tubes.
Yeah, I think one of the things, from an investor perspective, that makes it really exciting, as you highlighted, is that even a single cell type, has a massive market potential here. How do you kind of view that trade-off internally around continuing to progress the platform and go to kind of later and later stages and unlock more cell types, versus focusing on commercialization of the existing cell types?
Yeah, that's a great question. It's a question that we get quite a lot, but our focus is mainly about the platform and moving it forward. So we know we specialize in development stem cells and developmental, trajectories. So we know what we know, and we've collaborated with the right people, like Immune and other partners that help us take our hematopoietic stem cell product further. And of course, for the other cell type, within endothelial cells, we have a great partner here in Israel, and we have a lot of different partners for different cell types. So, yeah, that's how we're moving it forward to get the experts from every field.
Fantastic. Thanks so much, Ohad. So next up, we're going
Thank you.
over to the world of gene therapies with Matt from Ocean Bio.